Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, faculty at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today, I am going to discuss with you about the aspect of sample preparation for proteomic experiments. Under this particular paper, Techniques in Molecular Biophysics. At the end of this particular module, you would be able to understand how important it is to understand proteomic platform for research. Proteomics from a clinical perspective, patient phenotypes and health-related research questions, about aspects pertaining to patient samples, the input in proteomic experiments, the necessity of homogeneity of samples that come across in the aspect of proteomics, biomarkers and their tissue source, isolation of proteins from tissue samples, and also the important aspect about the high abundant proteins, doing proteomics and how salt affects the resolution of gels, protein isolation and solubilization, and dealing with hydrophobic proteins that you may come across at the time of protein isolation, gel resolution. So students, when we talk about proteomic experiments, you understand that we are going to talk about aspects of identifying biomarkers, identifying drug targets, understanding differentially expressed proteins, and you could also be talking about protein profiling of a particular tissue. But in spite of all the sophistication that is there with regard to mass spectrometric analysis and protein identification, it becomes very important to have a good proteomic sample for processing. This has a direct implication on the quality of experiments, the flow of experiments, and finally the accuracy with which you are addressing a particular research question. So let us see what is it that we can do to have a good proteomic sample. Time course of medical research. From the very beginning, when Mendelian experiments confirmed the concept of inheritance, and subsequently when the DNA structure was solved by Watson and Crick, genomics has come into emergence. Large-scale DNA sequencing has provided vast nucleotide sequence data that builds the genome of various living organisms like plants and animals, including man. The Human Genome Project has been drafted and most of the genome content is been analyzed in detail. However, it fails in certain aspects pertaining to the functional outcomes in cells and tissues. This in certain ways makes genomics static in nature. It is thereby very important to understand regulation of genes and the understanding of the quantitative and qualitative amounts of messenger RNA, which is more a realistic way of understanding gene regulation and their regulatory functions. This helped in the emergence of transcriptomics. It can be noted that gene microarrays provide snapshot of the expression of many of these messenger RNA in the cell. However, messenger RNA levels may fluctuate and are not a reliable reflection of the different proteins that are expressed in the cell. This may arise due to difference in stability of RNA or in the variation in the extent of translation because of alternative splicing. Proteomics then emerged and was coined to complement genomics and is a field that looks to understand protein profiles in various organisms, cells, and tissues. The application on human body in various pathophysiological states helps to understand molecular mechanisms and their outcomes in terms of clinical scenarios. This holds a lot of scope in health and medicine. Even as we understand the aspect of proteomics, you go on to understand that encompasses of understanding the structure, function, expression, and phylogeny of this particular proteins. But from the perspective of regulation, we understand proteomics from the expression point of view. Further, levels of protein may change due to folding, misfolding, post-translational modifications, and protein stability. And even as we speak and understand proteomics, there emerges a new concept called as interactomics, which discusses or delineates the different interactions that possibly exist between the expressed proteins with other proteins and DNA in the body or the cell. Proteomic platform of research. Proteome is a portmanteau of protein and genome as per Mark Wilkins, who coined this particular word proteome in the year 1994. A proteome is a collection of proteins of an organism's tissue at a particular point of time under a set physiological conditions. Proteomic profiling is an experimental platform to map all the proteins expressed in a given phenotype, and the differential expression compares the protein profile in two or more phenotypes. In the process of compilation of a proteome, proteins are identified and interactions between different set of proteins and other biomolecules is studied. Such interactions constitute different metabolic processes that occur in cells at all times. It is a comprehensive analysis of proteins in different cell types under various physiological conditions. And you could also be studying the associated post-translational modifications related to different functions and the changes in expression pattern with time and environmental conditions, genetic factors, and finally the disease pathology. Proteomics is an extremely interesting field because of the buildup of a complete proteome and the study of all the above discussed goals that has not been complete even for a single cell. Hence, there is immense scope to unravel the complex world of proteins, and each new study opens a new horizon waiting to be discovered. This is also encapsulate the fact that researchers across the world are analyzing subproteomes and by selecting a set of cells and particular conditions. This is done in order to understand the more complex picture of proteomics and the dynamics involved. And when these become relevant to human body and health, it is termed as clinical proteomics. Clinical proteomics comprises of protein profiling, which could be done either by gel-based proteomics or non-gel based proteomics. However, both these techniques require a very disciplined approach to sample preparation, which is a key to a successful proteomic experiment. Some of the ways and means of effective sample processing for gel-based proteomics will be presented here. So, even before we start, let us try to understand how proteomics is relevant from a clinical perspective. So, you could be doing a biomarker discovery, that is for the diagnosis of a disease. You could be identifying a new drug target for which you would be developing certain ligands or drug molecules. Diagnosis of variously closely mimicking disease phenotypes which you need to differentiate for prognosis and the right therapeutics. You need to understand the patient drug response. You could be monitoring prognosis of a disease. You could be evaluating the therapeutic efficacy of a particular drug. You can determine the cancer staging for efficient therapy, as different stages have different prognosis and treatment. You could also be complementing and validating all the other experimental outcomes in the field of genomics and transcriptomic analysis. You can correlate the protein expression with the pathophysiological conditions. And finally, as you understand, today there is a consortium to understand all the proteins that are expressed in the human body, and you could be adding data to this particular proteomic bank. Gel-based proteomics and its basic principles. Proteins are isolated from the body tissue samples and are profiled on acrylamide gels based on the intrinsic properties of charge and molecular weight. These protein profiles from two different phenotypes are compared to identify differentially expressed proteins. The qualitative variation among the proteins are linked to the clinical scenarios. Consistency of the expression across a number of biological and experimental replicates confirms the association of this differentially expressed proteins with that particular clinical phenotype. Lastly, the validation experiments on a larger cohort from different geographical and ethnic populations helps to translate this particular discovery that can be used by clinicians in the hospitals. Body tissue samples, the complexity in proteomic experiments. As we all know, proteins are present in all tissues of the body. They could be present in organ tissues, they are present in serum, cerebrospinal fluid, pleural effusion, ascitic fluid, synovial fluid, urine, saliva, and even tears. Therefore, when we set out to do a proteomic experiments, tissues are selected based on the feasibility of access, ethical issues concerned, the utility value to a clinician as a translational product, and finally, proximity of the tissue to the site of pathology. Organ tissues have lesser number of proteins and therefore easier for a researcher to analyze a handful of differentially expressed proteins. Whereas serum or even the plasma is a systemic circulation tissue and represents a lot of proteins, and it becomes that much more difficult to identify certain biomarkers in a systemic circulation which represents all the possible pathological and physiological events that could possibly be taking place in the body, but it is that much more easily accessible. Therefore, the bargain between the two would be to do research using fluids in the vicinity of the organs that are more reflective of the pathophysiological events and need semi-invasive techniques. So, as we see in the figure, for the clinicians, they would want the biomarkers to be in the serum, whereas for the researcher, identifying of the biomarker is that much more easy on the organ tissue, akin to finding a needle among a few straws of hay. To actually identify proteins, you have a lot of research questions that need to be answered, and you need to use the right body tissue source. Now, let us take the examples of the research questions that exist in the clinical side and what possible tissues that we could be using as proteomic samples. Differentiation of variants of advanced ovarian cancer. Here, you need to differentiate primary ovarian cancer from metastatic ovarian cancer, and the advantage of both the patients presenting with ascitic fluid helps to use ascitic fluid as a potential source. Next, identification of biomarkers in leishmaniasis. So, you could be using serum that makes it very easy for the clinician to use at the OPD. Identification of drug targets in tubercular meningitis. As you understand, cerebrospinal fluid is a fluid that covers the brain and is in a very close proximity, and this helps to understand the events that are there in the brain. So also, if you need to identify newer biomarkers for a condition such as Alzheimer's disease, which today is very difficult to diagnose, you could be using CSF. Understanding the response of a particular cancer, say for example ovarian cancer tissue to chemotherapy, you could be using the ovarian cancer tissue, as you have a protocol of a surgery followed by chemotherapy. So it means you have access to this particular sample or the cancer tissue. Next, differentiating Crohn's disease from intestinal tuberculosis, which is a very big question and an important research aspect in gastroenterology, and for this you could be looking at the intestinal mucosa. Finally, if you need to understand the pathogenesis of a particular parasite, for example Cryptosporidium parvum, you could be using the parasite cells to understand what are the differentially expressed proteins or what are the possible interactions that these proteins have with the human cells in order to cause a disease. Let us now discuss the aspect of availability of samples and scope for tissue banks. Translational research in health sciences depends a lot on the detailed study of human tissue samples. High recruitment aiding a successful clinical trial should be taken as a clue for tissue procurement and easing translational research. A national tissue bank bound by ethical, legal, and medical guidelines is necessary to coordinate tissue collection and clinical data storage. Standard workflows and operating procedures for the collection of fresh frozen tissue samples can be worked out. Sometimes procuring the right samples in sufficient quantities is the main bottleneck for conducting a good clinical proteomic experiments. However, the availability of a good tissue bank has its own advantages. 1. Less time would be spent on procuring the tissue samples. 2. Validated results are obtained sooner. And 3. Variation-based studies could be easily analyzed on retrospectively collected sample tissues. Homogeneity of proteomic samples for good quality gels. Successful identification of a biomarker or a drug target is based on the presence of a protein spot in a consistent number of biological replicates. This implies that there is a requirement of homogeneous set of patient samples that are to be compared. Inputs from the clinician are vital in drafting the inclusion and exclusion criteria for patients. Stringent protocols have to be followed for sample collection, its storage, and processing. The one-dimensional and two-dimensional run parameters have to nearly be the same to procure replicate gels. Homogeneity of the samples in all these ways helps to enhance the statistical significance of discovery and eliminate confounding factors. As we see in the figure, homogeneous samples have been used to run 3 experimental two-dimensional gels. Because of the homogeneity of the samples, we see a consistent pattern in the expression profile of proteins in all these 3 gels A1, A2 and A3. Even as we talk about proteomic analysis, let us try to understand what is a good control. Before that, let us understand how proteomics works. Proteomics essentially comprises of comparing protein profiles between different conditions, and we associate the differential expression of proteins with that particular disease phenotype. Therefore, this requires a very good selection of control in order to say what the differentially expressed proteins are. A definition of a control varies from research question, and this needs to be understood. For example, if we need to identify a biomarker for Alzheimer's disease, we would be comparing the protein profile of the disease with the normal healthy control. If we talk about biomarker for leishmaniasis, we again would be comparing the protein profile from the diseased with the protein profile from a healthy, normal individual. Next, if we need to talk about the treatment outcomes in cancer, we would then compare the proteins isolated from the diseased responders with the non-responders diseased patients. If you need to understand cancer progression, you would then compare stage 1 disease with a stage 4 disease. Then finally, if you want to differentiate two particular variants of cancer, then you would compare the primary disease with the metastatic disease. It is important here to understand that tissue procured from a site slightly away from the infected or the cancer site cannot be considered as a normal healthy control, as we are unaware of the molecular events that could have started in this particular normal tissue. High abundant proteins. Fluid tissues like serum, ascitic fluid, pleural effusion, and cerebrospinal fluid contain a high percentage of proteins expressed in micromolar range. These proteins are called as high abundant proteins. High abundant proteins hinder the detection of low abundant proteins. This reflects when there is more noise as compared to the signals that are obtained on the gel pattern. What are some of the high abundant proteins in these fluids? You have albumin, immunoglobulins, transferrin, fibrinogen, alpha-2 macroglobulin, alpha-1 antitrypsin, complement and hepatoglobin. And in the moderately abundant protein category, you could have apolipoproteins, alpha-1 acid glycoprotein, lipoproteins, complement factors, ceruloplasmin, and other complement factors. We therefore understand that almost a very small fraction of this particular protein profile comprises of the low abundant proteins. We therefore need effective devices and strategies to remove out a bulk of these high abundant proteins in order to be analyzing the low abundant proteins. Affinity chromatography as a method to remove high abundant proteins. High abundant proteins can be selectively be removed by antibody based affinity chromatography to enrich other low abundant proteins. The methodology is very simple and is illustrated in the figure below. You have the crude human serum or the plasma that is loaded onto a multiple affinity removal column that has a lot of antibodies against the high abundant proteins. When you remove out the flow and elute this particular column, you will obtain the low abundant proteins that are ideal for your clinical proteomic experiment. Just to have an understanding of the difference between the raw loaded material and the elute, we have shown the picture of a SDS page on the right side. As you can see, the raw serum has a certain number of proteins on lane 6 and lane 7. On the other hand, you have a higher representation of proteins in the elute that are run on lanes 4 and 5. That is because you have a removal of the high abundant proteins, and because of the removal of this particular noise, you are able to now have a better resolution and the appearance of these proteins as bands for protein profiling. Protein enrichment technology. In order to have a good proteomic sample, it is important to use certain technologies that will enhance the quality of the proteins that you would be using for your particular experiment. And there are certain columns that are available for this particular purpose. The protein enrichment technology decreases the dynamic range of the protein concentration in the sample by employing a large and highly diverse based library of combinatorial peptides, which dilutes high abundant proteins and concentrates the low abundant proteins. So in the figure, when you see, you understand that you load a particular protein, there is a binding of these proteins to these library of peptides as you see in the middle column, and when you finally elute out the proteins that are bound to these ligands, you would have essentially decreased the dynamic concentrations of the proteins. Is it really effective? Yes. As you can see from the gels below, you see a gel on the left hand side which has the raw sample. You see a gel on the right side which is the elute after processing it through this particular gel or protein enrichment technology, and you find that there is an increase in the number of spots on the right side. You also find that there is a better resolution to these spots as compared to the one in the left. Thereby, you go on to understand that there are more chances of identifying low abundant proteins using this particular technology. Also, because you have certain proteins that are bound to the high abundant proteins which could be having a housekeeping function, they too could be represented on this particular protein profile snapshot. Thereby, you could be having a differentially expressed proteins and you could be identifying them with that much more better accuracy. Selective protein enrichment based on function. We have seen how we could be improving the quality of the proteomic sample by affinity chromatography to remove out high abundant and moderately abundant proteins. Proteins could also be subjected to a wide variety of chemical modifications after translation called as post-translational modifications, which are crucial for the protein's function. For example, phosphorylation, which is an integral part of many enzymes and structural proteins in the process of cell signaling, are implicated in cancer. Therefore, selective enrichment of certain proteins like phosphorylated proteins helps in a better understanding of signaling pathways that are triggered in cancer. Likewise, glycosylated proteins could be studied in detail in the field of hepatitis. Acetylation, summation of nuclear receptors could be studied in breast cancer and prostate cancers. This helps to improve the efficiency of proteomic experiments based on the study and analysis of proteins that are implicated in the pathology and disease. Sample processing for gel electrophoresis. The whole idea here is to have a good representation of proteins in a particular tissue. For this, we would need to break covalent and non-covalent interactions that could possibly be present between proteins so that you are able to separate out the polypeptide chains into smaller subunits which are represented on the gel. You need to keep in mind that these proteins are intact by using protease inhibitor cocktail. You should be preventing artifactual modifications of polypeptides. Another important thing is to remove substances such as salt, DNA, RNA, carbohydrate, and other possible ionic molecules that interfere with the process of electrophoresis. You also need to keep the proteins in a solubilized state during the entire period of electrophoresis. You need to maximize the proteins of interest in the sample and at the same time minimize the high abundant protein fraction in the sample. It becomes very important to maintain homogeneity of the samples by proper inclusion and exclusion criteria at the time of patient screening. You need to procure the tissue at the right time, and you also need to preserve and keep the proteins at the right conditions for the particular experiment. Protein isolation and solubilization using detergents. Organ tissues are dissected neatly. The tissues are washed of blood, debris, slough, and mucus. The tissues are cut, minced, and homogenized in a solubilization buffer. This particular buffer or the solubilization buffer comprises of strong detergents and denaturants. This comprises of 8 molar urea, 2 molar thiourea, and 4% chaps. These reagents are either neutral or zwitterionic, meaning that they do not impart any charge to the protein. 8 molar urea is a very strong detergent and it helps by perturbing water, thereby preloading the solvent to accept nonpolar groups by subtle disruption of water's preferred structure and reorients the water around polar atoms. 2 molar thiourea helps in solubilization of certain hydrophobic proteins, and chaps is a detergent that helps in the solubilization of membrane proteins. Effectively, the combination of these three detergents helps to disrupt the interactions that stabilize protein structures and thereby makes it that much more easy to solubilize the proteins. However, this particular lysis buffer helps to solubilize the proteins but preserves the charge on the proteins that is so very essential for isoelectric focusing. Proteomics can be done, but there are a few troubleshooting points, especially from the aspect of salt. So let us try to understand how we can understand the salt contamination. Before that, let's just go through the effect and result of high ionic content on protein electrophoresis. You could be having precipitation of proteins that causes streaking in the gels. There could be an increased conductivity of current that results in heating and burning of IPG strips. You could have movement of water because of osmosis, which results in drying and swelling of the strips. There could also be a creation of a salt font which terminates isoelectric focusing. We at our laboratory clinical proteomics facility have developed a simple cost-effective technique based on the fact that ionic impurities conduct more electricity. Therefore, the salt concentration can be determined by the extent of conductivity of electricity by that particular sample. As you see in the graph, more the ionic impurities, more is the conductivity, which reflects on the quality of the sample. The advantages in knowing salt contamination is enormous because there is precious nature and paucity of the clinical samples. You have a lot of effort and time that is going into procuring and processing these samples. The high costs that are involved in the consumables used in two-dimensional electrophoresis. And finally, understanding contamination is important because good separation can never be achieved with the contaminants. Now, having understood that you have salt contamination, you could possibly take the sample and you could be doing dialysis, gel filtration or precipitation methods to remove out this particular ionic contaminants. Lipid and two-dimensional electrophoresis. Lipid is considered a contaminant in tissue samples in clinical proteomics. While even low lipid concentration distorts the resolution and visualization of protein bands on SDS page, higher amounts of lipid have been shown to have minimal effect on the resolution of spots in two-dimensional gel electrophoresis. The most abundant lipid is a phosphatidylcholine, which is a neutral lipid. It has an electric dipole moment of 10d, and this results in protonation of negatively charged phosphate groups at the acidic end of the strip to give the lipid a net positive charge that results in burning of the acidic end of the strip. The burning is clearly demarcated by a thick vertical streaking on the left part of the gel, and the length of the strip lost is seen to be proportional to the amount of lipid loaded. This has effectively been shown by experiments conducted in our lab. The four gels A, B, C and D show the gels that have different and a gradual increase in the concentration of the lipid, and as you can see, distortion is more in gel D with a higher concentration of lipid as compared to the other gels. Hydrophobic proteins is a real phobia for researchers in proteomic experiments. Hydrophobic proteins are proteins that are located on plasma membranes and other cellular membranes. Hydrophobicity is caused by the presence of one of the differently long hydrophobic domains, and their high degree of glycosylation along with phosphorylation causes difficulties in electrophoretic separation process. Limited or sometimes complete insolubility of these hydrophobic proteins, such as those of integral membrane proteins in the pure aqueous media, interferes with proteolytic digestion. Sometimes Triton X100 and CHAPS have been used to solubilize and prevent aggregation of hydrophobic proteins. Detergent lipid protein ratio is a very important factor for successful solubilization of membrane proteins. Mild concentration of detergent, which can strike a balance between maximizing the solubilization yet preserving the intrinsic charge on the protein for IEF, is the best bargain for effective procurement of proteomic tissue samples. In protein electrophoresis, when you do two-dimensional electrophoresis or differential in gel expression, it becomes important to have a hold on resolution of the spots. What do you mean by resolution? Resolution of a gel is the ability to differentiate one particular protein spot from another. It has a lot of advantages. What are these advantages? One, better the differential analysis that would be required for either biomarker discovery or identification or a drug target. Second, you would have a better mass spec analysis of the identified protein. There are lesser chances of false positives. You will have a better relative quantification when you say that one particular protein has that many fold times upregulation or a downregulation. You also would have better statistical significance with respect to the experimental outcomes. And finally, you would have a better efficiency of biomarker discovery which associates the protein expression to a clinical phenotype. Finally, what have we understood in this particular module? Let us summarize. Sample preparation is a very important and integral step in the workflow of proteomics. Patient screening, sample collection, and tissue banking are important pillars of clinical proteomics. Experimental conditions can vary and need to be standardized based upon the research question. Salt, lipid, high abundant proteins, hydrophobic proteins affect the gel electrophoresis. Protein solubilization is vital for good isoelectric focusing and two-dimensional electrophoretic separation and is accomplished using an array of detergents and chatropic agents. A good gel is one which has a lot of protein spots with a very good resolution.